This UCSD TV program is a presentation of University of California Television for educational and non-commercial use only.
We all strive to live a long, prosperous, and healthy life. With advances in health and medical sciences, this goal is ever more attainable. The Sam and Rose Stein Institute for Research on Aging is a non-profit organized research unit under the auspices of the University of California at San Diego, committed to advancing lifelong health and independence through research, education, and patient care. To better empower and improve the lives of young and old alike, the Stein Institute presents the following program.
Our speaker tonight, Dr. Richard Schwab, tripped on my own words there. Dr. Schwab did his undergraduate training at UC Berkeley. He received his MD from Albert Einstein Medical School, and he did his subsequent clinical training in internal medicine and hematology oncology here at UCSD through a residency and fellowship. He has joined the faculty in the Cancer Center and is going to talk tonight about individualized cancer care. He does research on identifying biomarkers related to breast cancer and other cancers, as he pointed out. And by identification of biomarkers helps to develop better diagnosis and treatment of different types of cancers that affect all people. He also has a lab that collects samples, so it's a biorepository to help others find biomarkers in the treatment of cancer as well. So without further ado, I'd like to introduce our distinguished speaker who has received grant funding through the NIH and private agencies and published in some of the most renowned medical journals today, including the Proceedings of the National Academy of Science and Nature Genetics. Dr. Schwab, thank you.
Well, thank you for that introduction, and I'd like to thank the organizers for the opportunity to speak tonight. As you heard, I'll be talking about individualizing cancer care. And the truth is, it's easy to get distracted by terminology. This concept of individualizing care or tailoring treatments for patients individually is really just another way of saying modern medicine. This goes back to the earliest days of medicine, where physicians used some of the early microscopes to look at tumors and to define, for example, what is cancer. I'm going to start by giving an overview of cancer and the biomarker research, which I think fits into this model of individualizing care, which again is just a history of modern medicine. And then I'll give some examples, including estrogen receptor and HER2. Obviously, my clinical work is focused on breast cancer, and some of my research focuses on breast cancer as well. So throughout the lecture, I'll be leaning back on some of those examples, although the biomarkers I study do extend certainly beyond breast cancer. And then I'm going to talk, I decided to, in terms of future biomarker development, I thought I would talk about sequencing, just because the technology is moving so quickly and there's so much opportunity in that area. I certainly do research in other areas, but I thought that that would be the most interesting area to discuss, and certainly the pace of advances in sequencing is unrivaled in any other area of modern medicine. So to start at the beginning, what in the world is cancer? So this is the definition is a group of cells with uncontrolled growth. So that initial piece is just a tumor, and then it has to have additional characteristics, either invading adjacent tissues and or spreading to other sites through the blood or lymphatics, and that would be metastasis. And this seems like a fairly straightforward definition, but it really isn't. This is an incredibly heterogeneous set of diseases. A tumor, a cancer that only invades adjacent tissues and never metastasizes is usually cured with surgery. So those cancers are the good cancers. Unfortunately, that's not the majority of what we see in cancer, and cancer mortality is mainly determined by metastatic disease, particularly by metastatic spread through the blood. We certainly hope to be able to develop treatments for metastatic cancers, and there have been successes where we can cure patients with metastatic cancer, testicular cancer being a good example, and also hematologic malignancies like leukemia and lymphoma. But based on the last 40 years of cancer research, it seems more likely that prevention and early diagnosis will lead to cures as opposed to treating patients once they already have metastatic disease. Now, progress in cancer has been slow, and I put this slide up to remind us all that the advances in heart disease really have set a very high bar for expectations, and cancer has definitely lagged way behind. So I think it's important to put this early in any lecture about cancer, because there's been a long, unfortunate tradition of over-promising and under-delivering in cancer research, and I think it's important to be realistic. Especially as a young investigator, I need to be realistic about what can be achieved. The National Cancer Act was passed slightly before I was born in 1971, and compared to heart disease, you can see that very little progress has been made. So back in 1950, we have the rate of heart disease absolutely dwarfing death due to heart disease in the United States, absolutely dwarfing the mortality rate from cancer. But by 2004, the advances in the treatment of heart disease had dropped the mortality rate dramatically, whereas age-adjusted death rates from cancer had declined only modestly. When you get rid of that age adjustment, the rate of cancer has actually, the number of deaths from cancer have actually increased over time. So we've made some progress, but it's been very modest. So the big question is, what are we doing wrong? And the answer may be nothing. One of the problems we face is that heart disease seems to be primarily a single disease. This is mainly obstruction of the coronary arteries, and controlling blood pressure, cholesterol, improving diet and exercise has made huge improvements. Cancer is an incredibly heterogeneous set of diseases. The initial definition of what cancer is started by site of origin. So breast cancer is one disease, a cancer that arises in the lung is another. And then as we've gone forward and learned more about cancer, we've split these diseases up into smaller and smaller groups. So breast cancer is divided into groups based upon both the histology, so what the cells look like under a microscope, but also based upon molecular differences that have been found over time. We keep finding more and more molecular differences, and we end up with more and more diseases. It seems impractical at best and impossible at worst that we're going to develop cures for these thousands or maybe even tens of thousands of different diseases. So from my perspective, one of the goals of biomarker research is to try to find these changes, these molecular changes that are common between different cancer types, so that we can, at least in some cases, stop thinking about a particularly molecularly defined breast cancer and instead think about a particularly molecularly defined cancer in general that could be treated the same way regardless of the site of origin. And this makes a lot of sense. Cancer is fundamentally a broken cell. The cells are deranged, and they're just growing out of control, and there's only a restricted set of proteins and signaling pathways that can be broken. So regardless of where these cells are when they take on this malignant phenotype, it's likely that they're going to be relying on some common pathways that we can identify and target. So biomarker research is essentially another, I think, in this case, useful term to describe modern medicine. This is a way of categorizing essentially all of diagnostic medicine. So on the left-hand side of the screen, I've listed out these different categories of biomarkers, and I think they're useful to look at. And then on the right-hand side, I've listed out some of the different molecules that can be useful for molecular biomarkers. Biomarker is a very general term for essentially any test that's used to meet any of the categories on the left-hand side that I'll be going through here. So there are physical findings that could be considered a biomarker depending on your definition. Imaging tests can be considered biomarkers, but for the purposes of this lecture, and I think for most of modern medicine going forward, the emphasis has been on these molecular biomarkers. Risk biomarkers are designed to detect high-risk people in a healthy population. These markers have been hard to come by, but when they've been found, they've been extremely useful. It turns out that some of these, again, depending on your definition, the most important risk biomarker for cancer is actually a cigarette in your mouth. So we know that cigarettes are the leading cause of cancer mortality around the world, and clearly targeting that biomarker is very important and beyond the scope of this lecture. But there are molecular risk biomarkers that have been found as well, in particular strains of HPV, which are causative for cervical cancer. Again, these types of risk biomarkers are very, very valuable to identify. And as we've seen with HPV, by vaccinating people against HPV, there is the promise of eradicating cervical cancer. Screening biomarkers are also very useful, detecting disease in a population. And again, cervical cancer really, again, is the gold standard. So Pap smears, which are widely available in the developed world, have dramatically reduced the rates of mortality from cervical cancer. But in the rest of the world, in the developing world in particular, where medical care is not readily available, cervical cancer remains a major killer. So obviously, while screening biomarkers are helpful and can reduce cancer mortality, they're harder to utilize because of the higher cost. And cervical cancer, again, is a great example for this. It's apropos that the Bill and Melinda Gates Foundation was mentioned, because one of the things that the Gates Foundation is funding is the dissemination of HPV vaccination in the developing world. And talk about your low-hanging fruit for reducing cancer mortality. The cost of screening every woman in the developing world every year with a Pap smear is extraordinary. But if we can actually vaccinate against HPV, I am very optimistic that the rates of cancer mortality around the world will decline from reduction in cervical cancer. It would be nice if we had a similar intervention that worked for cigarette smoking, but unfortunately there's no vaccine for that yet. Diagnostic biomarkers are interesting, but perhaps less valuable. This allows us to diagnose disease in a population which has signs or symptoms of the disease, and this is traditionally pathology. So a patient has a symptom. Our imaging tests are very good at identifying, in particular cancer, are good at identifying the location of a tumor. We can take a sample of that and look at it under the microscope and determine that it is in fact cancer. The next piece, though, the prognostic piece, is a place where there's a lot of opportunity for improvement. So a prognostic biomarker is used to determine the prognosis in the diseased population. In prostate cancer, for example, the majority of patients diagnosed with prostate cancer are not destined to die from their disease. Now, there is a large percentage of patients who are destined to die from their disease. And because prostate cancer is so common, it's a huge burden and critical to identify those patients. But because of this difference between the diagnostic biomarker and the lack of a prognostic biomarker in prostate cancer, we overtreat many, many patients. So there's this opposite side of the coin. We want to reduce cancer mortality, but we also want to reduce the burden of morbidity from unnecessary treatments. Prognostic biomarkers in breast cancer I won't be talking about in detail, but there has been some progress been made in this particular area. Breast cancer is also subject to this pseudo-disease phenomenon. A small but significant minority of patients diagnosed with breast cancer are destined to never have a symptom from their disease. So we have to be cognizant that as we develop better diagnoses, we have to readdress and try to improve our prognostic biomarkers, particularly for these quote-unquote good cancers, so that we can find those patients who really don't need aggressive treatment for their disease. For better or worse, this isn't an issue in many cancer types. Lung cancer, pancreatic cancer have a uniformly poor prognosis, and there really is no need for prognostic biomarkers in that population. Predictive biomarkers have been useful, and I will be giving HER2 as an example. I'll talk about that in more detail. But these are biomarkers that identify a disease subgroup that's sensitive, or in some cases insensitive to a particular therapy. So this can be very valuable as we identify molecular abnormalities in tumors that are predictive of specific targeted therapies against those markers. And then lastly, and again, perhaps less importantly, are response biomarkers, which are used to determine if a disease is responding after therapy. And for the most part, this is done with imaging. So a patient who has a cancer is receiving a therapy, and after a month or two on therapy, scans can be performed to see if the treatment's working. And there certainly is an opportunity to have biomarkers that would be more accessible, where we could get information about whether a therapy was working in a matter of days or weeks instead of having to wait a month or two. the right-hand side of the screen here, I think, is a useful review of the central dogma of molecular biology. So we have an example of a cell here, and the most robust piece of that cell is the DNA. So this is the inherited material that's present essentially uniformly in every cell in the body. And this DNA is used as the blueprint for the majority of the activities that happen in the cell. This blueprint can be copied into these working copies, which are called RNAs, which are not nearly as robust, but these are they use the same simple code, 4-letter code, as DNA. And then these RNA code is now then translated into protein. And for the most part, these proteins are the worker molecules. These are the molecules that are enzymes and structural components of a cell that really dictate what happens to a cell. And these proteins are used for manipulating carbohydrates and lipids, and while these are extremely important and very interesting biomarkers, I'm not going to talk about them tonight because it gets a little too complicated. One of the main advantages of DNA and RNA is that even though there's only a 4-letter code, and the physical properties of these molecules is very consistent. They're all negatively charged. Each of these molecules is similar in size. So when you have them, it's relatively easy to look at them. And that's sequencing, and I'm going to talk about sequencing in some detail. RNA, even though it's very fragile, is similar in that it only has these 4 letters, and they're very similar in terms of their physical properties, which makes looking at them in detail relatively easy. Protein is the exact opposite. It's a much more complicated and heterogeneous set of molecules, which there is no one simple way to look at. Anytime you want to look at a protein, you really have to develop a much more complicated assay, a much more complicated test, and it can take months, years, or even decades to figure out how to do that test optimally. And estrogen receptor, which is what I'll speak about next, is a really good example of how difficult it can be to look directly at proteins. So the bread and butter way that we look at proteins in clinical anatomic pathology is immunohistochemistry. And you can see here we have a tumor, and this can be sliced and looked under the microscope, and you can see the individual cells that make up the cancer. And if we want to look at the proteins in those cells, the most common way to do that is this immunohistochemistry assay. The most difficult part of this assay is this first step. What you need to do is you need to create an antibody that specifically recognizes that protein. And it's not just the protein as you might naturally find it. In order to process a sample for pathology, that sample has to be fixed so that it's hard enough to be cut. It has to be embedded in wax, and that makes significant changes to the protein. By the time it's on a slide and ready to be stained, that protein has undergone a lot of changes, and it's hard to even know what changes have happened. So we develop antibodies, typically in different animals that can make antibodies, and we can collect those antibodies and we can test them and see if they actually bind to the estrogen receptor. It's hard to even estimate how many different antibodies have been made and tested for the estrogen receptor. It wouldn't surprise me if the number went into the tens of thousands. And out of all of those, there are really only a handful of antibodies that robustly work on regular, fixed clinical samples. And literally, this has been a 3-decade process, and the College of American Pathology only released their guidelines for how the standardized protocol for how estrogen receptor immunohistochemistry should be done was only released last year in 2010. However, even with this somewhat variable assay, in the last few decades, tremendous progress in the treatment of breast cancer has been made, in part because of identifying the estrogen receptor. So we can identify the cancers that are estrogen receptor positive and need estrogen to grow. And we can see that these cancers have a very different prognosis. So even in the absence of therapy, this is the rate of survival in patients who develop metastatic breast cancer. And when you look, the estrogen receptor positive patients live significantly longer than estrogen receptor negative patients. And this is really an old finding going back to the mid-80s. So these two formerly, these estrogen receptor positive, estrogen receptor negative cancers would have been considered just breast cancer, and now we have 2 diseases where there previously was 1. And this process continues and continues as we split these cancers into smaller and smaller groups. Now, with the identification that estrogen receptor was present, we have tamoxifen, which was the first anti-estrogen therapy available. It binds to the estrogen receptor and prevents signaling. And this treatment, which has very minimal toxicity, certainly compared to chemotherapy, has a significant portends a significant improvement in survival. So patients who have lymph node negative breast cancer, who at 10 years would be expected to have a 71% chance of dying of their breast cancer, see a significant increase in survival up to 74.5%. And then for higher risk disease, where the cancer had already spread locally to the lymph nodes, a patient, instead of a 42% chance of dying from breast cancer, it's improved to 50%. So an 8% improvement in survival for what's a minimally toxic therapy. Now, that was an old but very robust example. Estrogen receptor positivity is very common. Approximately 60% of breast cancers need the estrogen receptor. So when you actually go back and look at that 8% and you start looking at a cancer like breast cancer, where 200,000 women a year develop it, you're looking at thousands and thousands of lives saved by identifying this receptor and targeting it. HER2 targeted therapy is a more recent but equally exciting advance. This HER2 protein is overexpressed in about 25% of breast cancer cases, and when it was first found, it was found to be associated with very poor prognosis. In a relatively short period of time from its identification, Herceptin, trastuzumab, is a therapeutic antibody that targets HER2, was developed, and has really flipped this disease upside down in terms of taking what was formerly a poor prognosis and making it what is essentially a good prognosis. For identifying HER2, we've had a similar problem. Immunohistochemistry, because of these many different antibodies and the fact that they're not reliable, isn't always conclusive. Fortunately for HER2, we have the luxury of going back and looking at the DNA. And that's because in most cases, this HER2 protein is present in too much quantity because the gene is actually amplified. For reasons that remain not entirely clear, cancers are able for the HER2 gene to make duplicate copies of this, and we can actually go back and look directly at the DNA. That's very nice because DNA, as I mentioned, is this very robust biomolecule. The currently used technique is called fluorescence in situ hybridization, and it takes advantage of the fact that DNA is actually present in two strands that complement each other. This is how DNA is replicated. The two strands will separate, and then the body can make a complementary copy to either strand, thereby yielding two identical copies of the DNA. We can take advantage of this and make a probe that contains the complementary sequence along with a fluorescent marker, and then we can apply this to the cells and see how many copies of the gene seem to be present. And here we have an example of this. So in this far right field, we see these individual breast cancer cells, and there should be two signals in each of these cells for the two HER2 genes. This is not a subtle example. If you go through and carefully count many cells, you'll see about 25 copies of the gene in each of these cells. The HER2 stain, when it works, does look very robust, but this has been unreliable in both directions. So the HER2 protein localizes to the plasma membrane, and that's what you see here, intense staining in the plasma membrane in a HER2 positive case. But these antibodies are not totally reliable, and we can have both false positives, where patients that are HER2 negative seem to have signal, and also HER2 positive patients where we don't pick up the signal for reasons that, again, remain unclear. And then we have another example of estrogen receptor staining, and it's nice to look at these side by side. The estrogen receptor lives in the nucleus, so the staining patterns look different. So it's very important to know where the protein's acting when you're looking at immunohistochemistry, since estrogen receptor in the cell membrane would have essentially no meaning, as opposed to HER2, where we expect it to be and where it acts and where it's important to target. Chemotherapy did make a big difference for HER2 positive patients. At the time we started introducing old-fashioned cytotoxic chemotherapy, which is essentially carefully given poison, we didn't know about HER2 positivity. But when we go back and look retrospectively, there is a huge benefit from giving chemotherapy to HER2 positive patients. At the time Herceptin was developed, patients who had surgically removed HER2-positive breast cancer would always receive chemotherapy and would have a much better prognosis than they did previously, with survivals in the 50 to 60% range. Much better than 20% certainly, but not where we want to be. This study here shows what happens when you add in Herceptin to that regimen. So when, in addition to giving chemotherapy, you target HER2, the survival, this is disease-free survival. So within a matter of 4 years, patients who received chemotherapy alone, fully a third of the patients had relapse with their disease. When you add in the trastuzumab Herceptin, only 85% of patients had relapse. So we're seeing, in a relatively short amount of follow-up, an 18% improvement. And even, again, at these early time points, we're seeing significant benefit in survival. So you can go back and look and do sort of a back-of-the-envelope calculation and see that the use of adjuvant Herceptin saves thousands of lives per year. So I think these are good examples of success stories in breast cancer, and they encourage us to look deeper. DNA is certainly not the only molecule to look at, nor is RNA, but because of the technological advances, it is becoming much more inexpensive to look at the DNA sequence and RNA sequences in cells in extreme detail. Given that HER2 can be identified in the genome, and we know about other mutations in the genome that are potentially targetable, I think that this is, because of the technological advances, really the most exciting area for biomarker discovery. And I'm going to talk about some of the projects that we have going. As a background, the Human Genome Working Draft was only published 10 years ago in 2001, and the first person to be resequenced only happened a few years ago in 2007. And I very specifically call it resequencing and not sequencing. The vast majority of sequence between any two people is absolutely identical. So when you resequence someone, nearly every single base pair will match the reference published sequence identically. There are relatively limited differences between people at the DNA level. They are important and we look at them, but this is also nice because we can start looking at cancer genomes. So we can look at the sequence in a cancer cell and look for any kind of mutation that might be present. The first cancer genome was published just a year later in 2008. And when you talk about publishing a genome or what is the first genome, it's actually a little bit subjective because cancers are heterogeneous. So not every cell has the exact same sequence. So how much coverage you need to get, how many times you need to sample the DNA and sequence and sequence and sequence to be sure you've captured all of those variations is non-trivial. And in none of these cases do we have 100% coverage where we can, with 100% confidence, call every single base pair in the human genome. In fact, the Human Genome Project finished, there remains regions of the genome that are very complicated and you can't reliably call the exact sequence. So while we know 99% or so of the genome, there still are these regions that are complicated. However, this, again, whole genome is sort of a standardized way of thinking about things, a sort of a typical amount of coverage where you could confidently call most of the DNA sequence in a person is now commercially available. And you can do resequencing for about $10,000. And that price is going to be dropping. In fact, if you look back over time, just over the last 10 years, the cost per genome has dropped at an unbelievable rate. So this white line is Moore's law. So this is the old law about computing power doubling every 18 months. And this is sort of the gold standard that all technology is held up against. Can anything ever beat Moore's law? Can any technology advance faster than Moore's law? And the answer is yes. So back in 2001, it would cost $100 million to sequence an entire genome. And right, you see that, okay, we're tracking along with Moore's law, and then all of a sudden there's this sudden drop-off. Well, this is called high-throughput sequencing or shotgun sequencing, where you take the DNA, you break it up into small pieces, and you put it out onto, there's a lot of companies that do this in different ways, but you put it out into an area, and you have a very high-resolution camera that looks at this. And then you can go, you make the complementary sequence, and each time a different nucleotide is added, whether it's an A, T, G, or C, you get a different color. So you keep doing this over time. So each little dot is flashing different colors, and you're collecting that information. And as the technology has gotten better and better, the cameras are better and better. Instead of looking at hundreds or thousands, you may be looking at millions of little dots. And you can build up this sequence of, even if you're only getting 50 base pairs for each little dot, you're getting this little fragment read of 50 base pairs. But if you have that for 200 million different pieces, you can line up all those little fragments and you can figure out someone's entire genome. So that's been the big breakthrough here. We seem to have hit a little plateau recently, probably as these high throughput technologies were more or less maxed out. But now we're moving on to these next generation sequencers, even beyond high throughput sequencing. There's a lot of these technologies being developed for different indications, but I will talk about one which I think has some novel features. Whether it turns out to be the near-term winner for how we do sequencing is a little hard to say, but it clearly has some potential advantages. So this is called ion torrent sequencing, and unlike the prior high-throughput sequencing, it uses pH instead of light to detect the different nucleotides that are being added. And this light detection is what makes the sequencers of today so expensive. A top-of-the-line high-throughput sequencing machine costs nearly $1 million, about $750,000. And then running the equipment is also very expensive. It's using these expensive reagents that emit light. This is a much more inexpensive machine. There's no fancy optics. It actually uses, it more or less co-opts this technology that's primarily used and has been developed for cameras called CMOS, and it lays on top of it all these little wells, and underneath it, it basically puts in pH meters. So we're in the very first generation of this was released last month, and as with all these technologies as it goes forward, we expect that these features, these microscopic features, will get smaller and smaller, so we'll get more and more sequence, and also the error rates, which is always an issue with these machines, should improve, which will be very important. So instead of a $750,000 machine, this machine might be in the order of $80,000 or $100,000. Running it will cost less, and the time it takes to run it is also much shorter. So instead of running the machine for a few days to get sequence, you can run it for a few hours and get sequence. And the way this looks is you have your template DNA in the wells, that's the red part here, and this works by sequencing by synthesis. So you flow in the different nucleotides, these A's and T's and G's and C's, and when the complementary base is present, you get incorporation of a nucleotide. That chemical reaction causes the release of one hydrogen atom. So one proton is released. But of course you can amplify up your template so that you get more hydrogens released, so you can have a little more signal. And then this individual microscopic pH meter can detect the release of that proton. So you flow over a T and you get a little bump. That means that in that well, the next base present was a T. If you get a double height bump, it means, oh, there were 2 T's in a row. And you can just go through this process, and you get a lot of data much more inexpensively. Now, regardless of what technology you use, there are limitations, significant clinical limitations to implementing sequencing. And the biggest one I've just decided to call garbage in, garbage out. So if you're not sequencing what you think you're sequencing, it's not useful. And cancers are very complicated. Tumors are actually often mainly normal cells. I mean, one of the characteristics of a cancer is that it's invading through normal tissue. So a lot of the cancers that we're most interested in studying actually percolate through normal tissue. So when you collect the tumor sample and you extract the DNA from it, well, you're mainly going to be sequencing normal DNA, or at least the normal appearing adjacent tissue, and not the tumor that you're most interested in. Now, there are a few ways to get around this. You can just sequence more and more, although even with inexpensive sequencing, that gets expensive. There is microdissection, where you can use a special microscope that has a laser that allows you to cut out the tumor cells. That's obviously very expensive, in terms of the cost of the machine, the cost of the labor to cut out all those samples. And if you need a lot of DNA to sequence, that means you need to cut out a lot of tumor cells. So even in a research setting, it's not terribly feasible, and in a clinical setting, it's essentially impossible. And then lastly, even if we had 1 million people working on these laser capture microdissection machines, in the process of clinical care, we frequently only get very small samples. If we're trying to figure out how to treat a patient who has metastatic cancer, and we do a biopsy and we get a tiny little piece, and we need part of it to look under the microscope and even decide if they have cancer, there's only a tiny bit left to do sequencing on. And if we have to slice it up and cut out just a few cells, there may not be enough material to sequence. So in the near term, a technology that I'm working on along with some real nucleic acid experts at our cancer center is termed ultra-deep targeted sequencing. So the first iteration of this technology, UDT-seq, was designed to focus in on that region of the genome where cancer mutations are found. As I've mentioned, most of sequencing is resequencing what's already known, and really not, quite frankly, not interesting. Most of the genome isn't even converted into RNA, isn't converted into protein, and even if we find abnormalities, we really don't know what to do with them. The targetable or actionable abnormalities in the genome are things that are going to affect proteins. So there has been a fair bit of sequencing already done on tumor samples. In the United States, there's the Cancer Genome Atlas, but there's really been quite a bit of worldwide effort on sequencing cancers. It's an obvious unmet need, and the technology is getting cheaper and cheaper by the day. So all of those mutations are actually entered into a database called the COSMIC database. So even though this sequencing approach only covers a small region of the genome, 86,000 bases, and the genome is 3 billion bases, just by focusing in on the right 86,000 base pairs, we can cover almost 90% of the cancer mutations that are known. The way this technology works is PCR, which is an amplification that's performed, again, taking advantage of the fact that DNA can be complementary, and you can focus in on small regions of the DNA and amplify them up, so you get lots of DNA of a particular region of interest. But this PCR reaction is done in water droplets in oil, these microscopic droplets, and I'll talk a little bit more about that. But what that technology enables us to do is run millions of these amplification PCR reactions in parallel in a single tube. So instead of running one PCR reaction for one site where you're looking for a mutation, we can run these PCR reactions across hundreds of hotspots that are known to have cancer mutations, cover dozens of genes that are known to be important in cancer, and then we can take these amplified DNA pieces and put them directly into all this fancy new sequencing. And in many ways, it doesn't even matter which sequencer we use. The cheapest, most accurate version is the one that makes sense. So perhaps there'll be ion torrent, perhaps there'll be an older technology. But with our first iteration, using some of the older high-throughput sequencing technology, we can get very, very deep sequencing, where we're sampling the same region of the DNA literally tens of thousands of times. So even if 99% of the DNA in the sample we're looking at is just normal DNA, or maybe from the adjacent tissue, or maybe it's cancer cells that don't happen to have the mutation. It may be that in a large cancer, there may be only a small fraction of the cancer cells that harbor a mutation that will make it resistant to the regular therapy and will eventually cause the cancer to relapse. Well, we can still pick that out because we're sequencing over and over and over, and we can detect mutations that are prevalent even down to 1% of that sample. And the nice thing about this technology, there are other approaches that use this kind of targeted approach, but most of those focus on just looking at known mutations. So there's a particular site in a particular gene that's known to have a consequence in cancer, so they do a very focused assay just for that one site, and they can only look at known mutations. With this technology, we're looking across all these hotspots so we can find those known mutations and also novel mutations. So just to describe in a little more detail how this PCR reaction works, you actually make these PCR reactions using primers. So these are complements to the DNA of interest that bind, and then you can use enzymes to extend that. And you run that over and over, and you amplify up the region of the DNA that you want to look at. But they can interfere with each other. So if you had all your different primers to amplify all these different regions in one mixture, it would never work. They would interfere with each other. So you put each of these pairs of primers into its own set of bubbles, and they're in separate little tubes, millions and millions of these bubbles. You take and you mix them together, so now you've got a mixture. The bubbles will not merge on their own in a tube. They're in oil, and they just don't come together on their own at that size. So now you've got a mixture of all these different separate PCR reactions. You can take your tumor DNA, You can break it up into small pieces and you can load it into similar droplets. And you put about 1/3 of a genome's worth of DNA into each of these droplets. And then using microfluidics, so this is some of this nanotechnology that you may have heard about, the two samples are brought together, and one-to-one the bubbles can be zapped and merged together. So you get a one-to-one mixture of your PCR primers and the genome you're trying to query, and now you've got millions and millions of these bubbles, and you can run your PCR reaction. You then break your bubbles, and you've got your amplified DNA of interest. And the way the primers are designed, you can actually design them so that they're ready to go straight into whichever machine you're going to be using. These machines do require a place to grab on a primer as well, and you can build that in. And this is very important if you're going to use this for patients. The goal is to get a biopsy and get that information back so you can use it for the patient in a timely manner. If you have to do a research assay and it's going to take 6 months to get the result, it's not going to be useful for the patient. So by designing this two-step process to work smoothly together, you can get results within a few days. And also by focusing in on these smaller regions of the genome, the analysis at the other end is also simplified. When you're looking across the entire genome, it's much more difficult to align all these sequences and figure out what's important. By focusing in on the regions of interest, it makes that analysis part more practical. Now, we've run some tests using this technology. We have samples in the Cancer Center biorepository, and we have different types of samples. So the very precious samples are primary tumors that patients have donated to us. And those samples we don't want to use for practice runs. We want to save those when we have something we know is working. They're expensive. There are not that many patients who are willing or able to donate samples. And when we get those samples, we freeze them and we store them in liquid nitrogen so that they'll be useful for many different purposes going forward. We also take small bits of those samples, and with patients' consent, we actually grow them up in animals so that we have what are called xenografts. So we have lots and lots of this tumor that we can work with. There are a lot of advantages to having those types of samples. You can test treatments against them, but you can also use them when you're trying to develop a new technology like this. We have a breast xenograft that we tested, and we actually found a mutation in a gene called HRAS. And what's interesting about this is this isn't known to be in breast cancer. This is a very, very common mutation in bladder cancer. So that was very exciting for me because this is a perfect example of a place where we may be able to take advantage of this common molecular derangement. There are drug companies developing agents to treat HRAS, to target HRAS. Of course, they're focused on bladder cancer and other cancers where this is a common target. But even if there's only 1 in 100 patients with breast cancer that has this mutation, if they're already developing drugs for it and we can identify the mutations, this is a great opportunity, in my opinion, to try to leverage that and make advances. The colon xenograft that we tested is much more complicated. It has a variety of mutations, including 2 different mutations in this APC gene that's well known in colon cancer, basically knocking out both of the genes. There's also a very common KRAS mutation that's actually clinically used, and this predicts a lack of responsiveness to a particular type of targeted therapy. So if we had been able to provide this data back to the patient in real time, this would have been very useful for their care. We have an ovarian case where there's actually 100% of the DNA shows a mutation in this very important tumor suppressor called P53. I suspect what actually happened in this case is not that both genes have the same mutation, but rather one of the genes is completely deleted, obviously not functioning, and then the other copy that's still present is mutated and not functioning normally. And then we also looked at a sarcoma case, and we actually found only rare mutations in these genes of interest. And it's not entirely clear what that means, whether this cancer harbors mutations that would be important to target, or maybe the bulk of the cancer really doesn't need these mutations to grow, and they may not be important to target. But that will be something that will be very important to sort out as we go forward. Now, once we have these results, an open question is, well, how relevant is this xenograft material to the real tumor? Well, for the breast and colon case, we had that primary tumor frozen. So we were able to go back and run the exact same assay on just this tiny piece of primary tumor, this precious tumor that we had saved. In the breast cancer case, you can see that this well, you can't read it, but this HRAS mutation was actually present in the primary tumor, which here is in dark red. essentially the same frequency as it was found in the xenograft sample. So in this case, the xenograft really held true, and it represented the same DNA mutations as was found in the xenograft that was grown later. The xenograft did have some less common mutations that seemed to be starting to come up, but they were not found in the primary tumor. So exactly what that means, again, is somewhat less clear. The colon case is more complicated. It had more mutations to begin with. The light blue shows all the mutations that are present in the xenograft. And when we look at the primary tumor, the vast majority of the mutations that are found in the xenograft are present in the primary tumor. Interestingly, one of the mutations that is found in the primary tumor was lost in the process of forming the xenograft. So this cancer had to learn how to grow in an inhospitable environment. And for whatever reason, that environment selected against the cells that had this mutation, and it selected for cells that had mutations in some other sites. So clearly, the xenografting process does change some of this DNA, the fundamental DNA nature of the cancer. It's important to know when we're using xenografts, but I think most importantly, it shows that this assay is working. So what are the pros and cons of this type of a targeted approach? Well, the pros, I think, are numerous. So the assay requirements are really minimized. We can use small samples. We can use heterogeneous samples. The DNA is very robust, so you can throw it in a refrigerator over the weekend and it's not going to change. And the cost of the assay, as envisioned using this new technology, is actually fairly reasonable. It's about $2,000, which is on par with some assays which we already use clinically in breast cancer. One of the drawbacks or cons of this approach is that from the Cancer Genome Atlas and other sequencing projects, we know that DNA mutations do not explain all of the diversity of cancer. There are other changes that happen in cancers. Now, why would we go after this first? Because we can. This is a robust assay. We can take advantage of this. We can find DNA biomarkers like HER2 that can be targeted, even though we know full well that we're going to have to move on and do additional work in other types of biomarkers to continue to make progress, RNA being an obvious next step. One of the other drawbacks of the current assay is it doesn't detect translocations, where one piece of a chromosome moves and becomes adjacent to another piece of a chromosome, and that can create fusions, and those fusion abnormal proteins actually can be very important in cancer. The assay can be changed to be able to detect those types of translocations, but that'll be a project in itself. I'm going to talk just briefly about things that we might do beyond targeted DNA sequencing. Again, using a xenograft model of, in this case, lung cancer, we were able to sort that into two populations, into a cancer stem cell-like population and a non-stem cell-like population. So one of the other advances being made in cancer is, again, recognizing the heterogeneity of tumors. Even within a given tumor, there are cells that take on stem cell-like features. We really don't quite understand what that means, but these cancer stem cells do seem to be resistant to chemotherapy. So they do, whether they're really stem cells or stem-like cells, the fact that they're resistant to chemotherapy means they're bad cells. They're worth knowing about and they're worth studying in terms of targeting. So we took these two populations, the lung cancer stem cell-like population and the non-stem cell-like population, and we did some basic DNA work using what are called SNP chips, where we look at the normal variations across the genome, and we also did whole transcriptome. So these samples had stable RNA because these are samples grown outside of the body, so we could keep the RNA, which is very fragile, in good condition, and we can use this advanced sequencing technology to sequence all of the RNAs. So rather than just detecting changes in the DNA, we can also pick up on changes that might have happened as that DNA is converted into these working templates, RNA. So when we look at the person's blood, their germline, we can see that their genome is essentially normal. The SNP array looks at the areas of the genome that are different between people. So most of the genome is the same, but this particular array looks at 1.1 million different SNPs. So these are 1.1 million base pairs that are frequently different between people. The other 2.9 billion base pairs are mainly the same. So it's nice, we can focus in on this. Now when you try to show this much information, it's actually a major challenge. So you can lay it out by chromosomes. So the biggest chromosome is number 1, 2, 3, 4, all the way around to 22, and then there's X and Y, and this is a male patient because we're seeing signal in Y. And the blue just shows how much signal we're getting. So if there was an amplification, the blue would go up, and if there's a deletion, the blue would go down. And then the two different types of, there's only A, T, G, and C, and in general there's just the two common versions of any given SNP. So there's the common and the less common type of sequence that one would have at a particular site, and that's shown in red. So if someone has two copies of the A allele, they're AA, they're up here. If they have two copies of B, they're down here, and if they're AB, they're in the middle. And you can see, as most people are, this gentleman is a normal mixture of those different variations. When we look at his tumor, we see something drastically different. So this is a lung cancer case, and we're looking at the stem cell-like population, and we see dramatic alterations in the genome. There are regions of amplification where the blue area is up, regions of deletion where the blue area is down, and when you look at these amplifications, you can see a split in this red area where there's AA, BB, and then there's an AAB and a BBA. So you can have three copies of the gene where there's an amplification, and you can pick up on that with this SNP chip. What was one of the more, and the 15 genomic alterations that we found actually matched up quite well with what's been seen in the literature. There's about 26 commonly seen genomic alterations in smoking-related cancer, so this smoking-related cancer matches what's seen. Now we look at the stem cell-like population and the non-stem cell-like population. According to the assumptions about these stem cells, we would expect that their DNA would be the same. The stem cells should be producing these non-stem cell-like daughters, as well as some of their own stem cells, but they should have the same DNA, and that's not what we found. We actually found that there's regions of amplification in the non-stem cell population, they're marked by green, that are missing in the stem cell population. And the way my hypothesis for this is that the stem cell population has turned on some of these stem-like pathways that help proliferation. That's a normal stem cell should proliferate, and it allows that cancer to grow without as many changes. The non-stem cell population is missing this boosting pathway and therefore needs another few mutations to compensate for that. Then we went further and we looked at the RNA. And to give you an idea of how much sequencing we did, we're talking about 40 gigabases. So this is a whole genome's worth. This would give you about 10x coverage of the human genome. This is the same as the Human Genome Program, which took a decade and cost billions of dollars, and in 2001 it would have cost $100 million. Well, now for a price that you can afford in a lab, you can run this much sequence. We can look at the two different samples at the RNA level, and we can start to tease out if there are differences in these RNAs. So what is an RNA? An RNA is a copy or a transcript from this germ, from the DNA. When we started the Human Genome Project, the assumption was there would be about 100,000 genes in the human genome. Well, when we got the results, we were very surprised. There are only a little bit over 25,000 genes in the human genome, basically the same as a mouse. Well, that's a little disappointing. We thought we were going to be a lot more complicated than mice. And it turns out that we are, but the way the body does this is through alternate splicing. So genes are not just a simple region of the DNA that gets copied and then produced into a protein. The regions of the DNA that are actually used to make protein, even for a given gene, are actually split up into little chunks called exons. And those little chunks are spliced together to make the actual RNA that's used for protein. Well, during evolution, it's hypothesized that this allowed for more accelerated evolution. These little chunks can be cut together in different ways, so you can get alternate transcripts. So where you have 25,000 different genes, maybe the gene starts at a different place, so you're missing some of the exons, and you would end up with different isoforms, maybe 38,000, and then the exons within the gene can also be left out, and then you end up with 75,000 different transcripts just based upon these RNA variations. So obviously this makes up a huge amount of diversity and may very well be important in cancer. And what we found was that, yes, in fact, this is important. If you just looked at the gene overall and how much of the gene was being expressed between the stem cell and the non-stem cell population, in this particular gene you wouldn't see a difference, this TIF1 beta. However, when you look, then this is the stem cell population up above and the non-stem cell population below. Red means that the transcript is decreased and how much is being expressed, and green means it's increased. And we see that this transcript, which has basically all the exons, is actually relatively low in the stem cell population, and an alternate transcript that's missing just a few pieces is elevated. In the non-stem cell population, it's the exact opposite. And we would have missed this if we weren't looking at this detail. So the full transcript is elevated. The one missing just a few copies, which we could call the stem cell-like transcript, is decreased. And there's actually another truncated transcript which is elevated. This particular gene is very interesting because it's been studied in other contexts, and it's known to regulate pluripotency and self-renewal in stem cells. So this may very well be the difference that we're seeing in this cancer stem cell population. And now what we're doing is going forward looking into other cancer types, looking at the stem versus non-stem cell populations to see if we can find common abnormalities that could be targeted to target cancer stem cells across different tumor types. So whether you're looking by UDT-seq at DNA or you're looking at RNA, we do have an issue, which is how are we going to get this information back to patients to treat them? These are all research results. They're run in labs which don't meet any of the criteria that clinical labs are required to meet in order to give a patient a result. So we have planned to develop a master clinical trial where we would plan and consent patients to return their research results. We've developed a plan where we have a committee that will decide what regions of the genome are interested to target, and also what results are returnable, what results are actionable and actually need to go back either for the patient's benefit or perhaps for their family's benefit. We're going to find patients who harbor recessive alleles in cystic fibrosis genes that they didn't know about, and it will be important for them to know about this so they can confirm it clinically, and it might make changes in terms of reducing problems that their offspring or grandchildren could have. However, we need to do these results in clinically certified labs, which means we have to bank samples in clinically certified ways so that when we go back and need to retest samples, they're available. So we've developed what unfortunately needs to be a fairly elaborate system where patients are consented. Their samples are banked in clinical lab. This upper box is all the clinical part of this. And then those samples can be released to the research group to do all the sequencing, to figure out which of these mutations are important to know. And if there is an actionable mutation found, we can then tell the patient's provider, who will then inform the patient that they generally have an abnormality that might be worth going after. Maybe something that would affect their reproductive choices, maybe something that would affect how they would be treated for their cancer, and give them the opportunity to say, okay, yes, I really do want you to go forward with this genetic testing. And if they do, then the clinical lab can log in, pull the complete report, know exactly what mutation has been found, and then relatively inexpensively confirm that clinically. They don't have to sequence the whole gene. They can just look for the exact mutation that's been found. So this is what we're trying to do going forward. At first, we'll try to use the least expensive assay, which for now is this UDT-seq assay. But going forward, we envision using this same type of approach even when we can afford to do whole transcriptome sequencing on patients' tumors. So with that, I will end it and thank the many collaborators who have been involved in this work. Kelly Fraser and Olivier Arizmendi in particular involved in UDT-seq, Christian Barrett involved in the sequencing, the RNA sequencing, and Wendy Wang involved in all of the sequencing. Karen Messer, our statistician, which is critical for looking at any of these results. We have a whole team of people involved in collecting samples and banking them. My mentor, Dennis Carson, who some of you may know since he was the former director of Stein and has many connections and knows everyone in San Diego. And also the clinicians who speak to their patients and discuss with them why it's so important to provide these samples that we can use to try to develop new biomarkers. And with that, I'll take any questions.